All right, so how is everyone?
Good.
Good.
How are you?
Excellent. All right.
So we're just going over this review paper, right?
I think so. I think Ryan had some other information he wanted to share related to ALS.
Hey, Nina.
Let's see. I think he just joined on, actually.
I did. Can you hear me?
Yeah.
All right. Excellent. So how are we doing? Sorry for the delay, everyone. I was just setting up here. I'm setting up over in Counterculture Labs. Let's see. Okay. So, did you give everyone...
Where is Counterculture Labs?
Well, actually, it's at Anand's house, so... It's in West Oakland, so... not quite yet a lab, so that's the yeah. So we have, I think we have GenSpace represented, we have BioCurious represented, we have Counterculture Labs, and we have actually Derek, and we have your Hackerspace too, which is in Vancouver, is that right? Or okay, I don't think Derek can.
Victoria.
Victoria. I forgot. And then we have Berkeley Biolabs as well. So we have a bunch of. And have I missed anyone?
Yep.
Yeah.
Let me see. Victoria, actually.
Victoria, yes, yeah. I think there's a little bit of a lag there, Derek, on your end.
I'm in Montreal.
Oh, hey, this is cool.
I have a distant GenSpace member.
Okay, hey guys from GenSpace. Yeah.
I'm at large.
Yeah, no, this is great. Yeah, so we have pretty much, we may actually have some people joining from Argentina and possibly India as well.
That would be awesome.
I got told, so.
Tonight?
What's that?
Tonight?
Yeah, tonight joining us. I'm not sure if they will or not, but they said they may.
Let's see how stable this is today.
Yeah.
Yeah.
It will really be transnational.
Yeah, so we have a bunch of people. I'm actually facing them right now. So we have a bunch of people here with us as well. I'm over at Anand's place in the living room. So yeah, I mean, I'd like to maybe just start off with, obviously you're all very familiar. Yeah, no, feel free to have a seat there. Yeah. Obviously you're all very familiar with the paper. Do any of you need the paper? If so, could you write it in, Nina, I think there's like a little text box, right? Could you write it in a little text box and send your email address over to Nina so she can send you the paper if you don't have it? So the paper is the Self-propagation of pathogenic protein aggregates in neurodegenerative diseases. And this is going to be much more of a discussion around the paper. It's kind of a timely topic, and it's also controversial as well because there are multiple hypotheses around the causes of neurodegenerative diseases. So just to give everyone a little bit of background on myself, before I kind of left and started doing the biotech startup thing, I actually helped most of the major biotech and pharmaceutical companies do drug development, and a large portion of the work that we used to do was actually CNS. So I worked with the Michael J. Fox Foundation on Parkinson's, looking at alpha-synuclein in the cerebral spinal fluid. We actually ran their human phase 1 studies and actually figured out how to actually run that and recruit those patient populations. I've also worked in Alzheimer's. Okay, yeah, we have a little background noise there. Also in Alzheimer's and ALS, where I was involved with actually Halpin Neurosciences. I was a co-founder of Halpin Neurosciences and the CEO of Halpin Neurosciences. We identified one therapeutic small molecule, actually, that we hope will potentially extend human lifespan or ALS patient lifespan. It's currently being tested in preclinical models, but we co-founded that. Les Halpin, unfortunately, who passed away, co-founded and funded that biotech startup alongside Eric Baller as well, who's my patient researcher. So he actually passed away at the weekend. Les was very involved in something called the Halpin protocol to get experimental access to drugs for patients that are experiencing life-threatening diseases. So that's kind of a little bit of background on myself and also on Les as well. Kind of want to dedicate this to Les, the discussion. Hopefully we'll continue to work on projects that will help us treat these diseases. And I also just want to mention something that was very notable today. So many of you have probably heard Lary Page announced that Calico was being formed out of Google, spun out of Google. So that is huge news for aging-associated diseases, which I think neurodegenerative diseases I would definitely put in that category. Art Levinson, the co-founder of Genentech, is actually going to be the CEO, which is huge news. So he basically ushered in the era of recombinant DNA and recombinant technology. And so they're going to be leading that forward. So it's a huge deal for longevity, aging, and obviously neurodegenerative diseases as well, which hopefully they'll tackle. There's still information coming through. Aubrey de Grey also wrote a little bit about that as well. So he's also included in Time Magazine. It was an exclusive in Time Magazine. So both Aubrey and Lary Page were mentioned there. I spoke with Aubrey a little bit earlier, and he's very excited. So he's actually flying back to the UK or somewhere else transatlantically. So, okay. So should we get started? Is everyone, Nina, have we got everyone basically ready with papers and things?
Yeah, I think so. I sent one out to Kelly, and that was it.
Okay, great.
Yeah.
So I'd like to kind of start everything off just really as a discussion. So is everyone comfortable with the importance of proteins and protein folding? Okay. So as we all know, proteins have to have a specific structure to do the function, and that's really, really important. If they're denatured, which is a term for misfolded protein, then they don't do what they're supposed to do. So this is what leads us to, we believe, many pathologies, including prion disease. So prion disease, if you've read the paper or heard of Creutzfeldt-Jakob's disease or various other, I think it's called scrapie in sheep as well. So the spongiform encephalitis, it's actually a disease that's caused by protein misfolding, and it's actually a terrible disease. The brain inflames in Creutzfeldt-Jakob's disease, and then eventually the person gets tremors, is unable to walk, and eventually they die. And that's all due to a normal protein being misfolded. So I think this is the premise of this paper, that really they're challenging us to look at protein misfolding as the possible causes of several neurodegenerative diseases. Either looking at, with the actual protein misfolding, either looking at toxic function or loss of function. So this is the two things that they actually classify. There's actually a great little pictorial, I think it's on figure 1, where you can actually see the areas of the brain affected by the different either external or internal kind of protein clusters or misfolding, sometimes plaques. And then you can also actually see a little pathology of what it actually looks like in the neuron or outside of the neuron as well. So they essentially cover 4 different types. A beta, which I'm sure many of you are familiar, which is involved in Alzheimer's. The tau neurofibrillary tangles as well, which is also potentially involved in Alzheimer's. Right now there are several competing theories in Alzheimer's. One is that it's as a result of A beta. The other one is that it's a result of tau. And some are that potentially it's as a result of both. And there are some other really interesting hypotheses around type 2 diabetes, and how, and another one is maybe A beta is protective. Maybe it happened after whatever goes wrong in the neurons that starts to kill the neurons occurs. So
I have a question.
Yeah. Mm-hmm.
Are there, and this is like my lack of prion knowledge, I guess, but are there proteins that typically end up in these aggregates? Like, are there specific amino acid sequences or basic structures that lend themselves to be more likely, like if expressed or overexpressed or not folded properly, then they wind up in these inclusion bodies or these deposits and lead to these different amyloids or, fibril, structures? I don't know if there's a specific amino acid sequence in common or something along those lines that people are trying to target.
Yeah, so they're repeat structures, and anyone that's particularly familiar with prions and the repeat structure that occurs there, please feel free to chime in. But it's in box 1. It actually covers the amyloid state of proteins as a framework to explain prion-like seeding. So it gives you an idea of how these repeat structures form and kind of build on each other. So they become oligomers essentially, and so eventually they can become plaques. And so they start at the microscopic level, and eventually they become large enough that we can see them.
So it's more based on, well, obviously sequence leads to structure, but I mean within these you would find a variety of amino acid sequences as long as they are forming these repeat structures.
Yes, yeah. So I don't think we have specific sequences that we know will always form prion-like diseases, but we do know that there's a tendency, if they have repeat structures and certain structures that can repeat, they can click and oligomerize.
Mm-hmm.
That's what we've seen, right, looking backwards. So what's interesting is, go ahead. Sounded like someone.
I think they discussed that on the top of page, on the right side of page 48 in the growing family of prion-like proteins. The first paragraph of that section, they talk about the aggregate clone stretches of amino acids known as prion-like domains, which occur in significant subgroup of RNA binding proteins.
Thank you.
We'll talk about it there.
Okay, thank you.
But I don't know what those domains are, but that's something that we could perhaps look into a little further as well, because that kind of links in with the therapeutic aspects too.
Yeah.
So kind of going through so there's the A beta, the tau, the alpha synucleon as well, which forms Lewy bodies as well. And now these are really interesting. So alpha synucleon and TDP-43 in ALS. So alpha synucleon is Parkinson's, TDP-43 is in ALS. These are both in the actual motor neuron themselves, which is different from what we see in Alzheimer's, which tends to be external to the actual neurons. Although there are some theories that the initial toxicity comes in due to a disruption of the microfibrils by the small oligomer units. And that actually kills the neuron, and then you start seeing these plaques that form afterwards. So that's an interesting way to see these pathologies is slightly different. Perhaps the toxicity is as a result of the smaller plaques, the microscopic plaques, or the oligomers. So I don't know if anyone has any points there to chime in on. So the prion-like properties, particularly the seeding, is very interesting. So what they found with prions is that it's very easy to seed. If you take a prion and insert it into a brain that or consume a prion, you're likely to get prion disease. So it's just a matter of time. These things are very, very difficult to break down. I've talked to friends of mine who've been involved in investigations around prion disease, and they treat it unlike any other disease we've seen, because any tools you use, it's very, very difficult to disinfect. So they actually just use the tools that they use just for prion-infected tissue, and then they throw it away if they need to use, if they need to investigate another disease, because it's just so infective and very hard to destroy. So the seeding tendencies are something that is likely, we feel, I guess the paper feels that is responsible for some of these neurodegenerative diseases as well. And also the structures can be highly polymorphic as well, so of the actual oligomers that form.
Ryan?
Yeah.
Yeah, a quick question. So the seeding concept then sounds like once you have a misfolded protein, it then causes additional misfolding?
That's right. Yeah.
But then it's the aggregation of those misfolded proteins that cause the pathology.
Exactly.
Okay.
And we know that that's true with prions. In the paper, I don't believe, and anyone who remembers this, please correct me. In the paper, I don't think they actually highlighted that we know that for neurodegenerative diseases, but I believe if this holds true, then if we were to infect someone's brain with a beta amyloid precursors, in theory, you'd also get beta amyloid plaques.
Okay. And how about the flip side? Are there certain types of proteins that don't cause misfolding of the same type of protein, but over time aggregate in a way that causes the same type of symptoms?
Yeah, actually, so this does occur.
It's kind of a non-seeding version of it.
Yeah. So this can occur. If you look at things like lysosomal storage disorder diseases, they don't cause any misfolding. They just fill the cells full of proteins, protein junk basically, and the cells can't break it down. So you do start to get like enlargement, particularly of the intracellular proteins. It causes enlargement of the cells, which can sometimes lead to death of cells as well.
Is that lipofuscin? Is that what you're talking about?
Yeah, lipofuscin, and there are also a lot of other lysosomal storage disorder diseases. So Gaucher's disease, Fabry's disease. We have therapies for these, and it's transformative what they can actually do. Mm. So it basically, you can take someone that has a mutation in one of these lysosomal storage enzymes, and you give them the enzyme, and they go from being very, very sick and dying very, very young to living almost a fully healthy life. I mean, the enzyme doesn't get everywhere, but it gets pretty much almost everywhere where it needs to go. But that's very different to what we see happening with these neurodegenerative diseases.
Ryan, I was just noticing when looking at these figures, I don't know much about prions, but like the kinetics of seeding, it's awfully similar to the kinetics of actin and tubulin polymerization. What's happening here. So when I looked at that, it looked very similar to that type of polymerization. Is there? I mean, clearly that's different, because that polymerization is influenced by ATP and ADP binding. So there's a hydrolysis of ATP, and this is just I guess the seeding is just taking place intrinsically. So it's not binding to any cofactor, right, to initiate the seeding or fall apart. It's just the polymerization of the misfolded parts to this prion protein here that's causing these kinetics to occur. Is that something that you're investigating? I mean, I guess are there differences, similarities between that sort of seeded polymerization?
Yeah, I mean, that's an interesting point. I mean, as far as I'm aware, we don't know that there are any cofactors, particularly with prion disease, but there may be. I mean, it's an interesting point, right? I mean, could there be other enzymes that are being hijacked for this purpose by the prions?
Yeah, because you get the same thing, right? There's like a critical concentration of let's say actin monomers or tubulin monomers, and below that concentration you get this. So if you look at figure box 2, for example, where they inject the mouse, or even the figure box 1b, where you have with seeds and spontaneous aggregation. That's pretty much essentially the same types of curves you get, although albeit much faster because this is happening in vivo. Mm-hmm. The same types of curves you get when you actually add in a fragment of F-actin, like filamentous actin, versus a monomer. Yeah. So I'm just wondering if there's any differences there, or any, I don't know if anybody's done a cross comparison between that and between this.
I haven't seen any. That obviously that doesn't mean that hasn't been done. But it's actually a really interesting point that you bring up, because if we're looking at treating some of these diseases, and we're looking at these kind of tangle type structures and these plaque type structures, which are oligomers, they're polymerized oligomers. Like, maybe it might be possible to disrupt them with enzymes that disrupt actin cytoskeletons or something like that, right? Or something similar.
Yeah, it's obviously the actin is a completely different, protein, but they do have small molecules that do disrupt that, or either support the filamentous structure like phalloidin, or disrupt it like the trunculin, which breaks apart or prevents lowers the critical concentration for seeding to actually happen. Yeah. Of course, that I doubt it would bind to this because it looks like it's a completely different secondary and tertiary structure, but I'm just wondering if the dynamics between all of these types of polymerizations and seedings were looked at with prions.
Yeah, I don't know the answer to that, so I don't know if anyone else has actually looked into that or has any feedback on that. But that's a really interesting idea, and that kind of leads to that interesting question. Could we find something that disrupts these polymers or these oligomerized polymers? I don't know. It's interesting, Oliver. Very interesting.
Can I just ask this for just a second? I'm really sorry.
Yeah, please do. Yeah.
I think you can keep going.
Oh, okay.
I think they're.
That was an internal discussion there, right?
Yeah.
A Gen space discussion going on. It sounded like Ellen, actually. So, okay. So infectivity is something that we've discussed a little bit on, and that was actually covered in the paper as well. And I guess the final point is really the therapeutic aspect. So I can speak a little bit to the therapeutic side of things. Didn't have a point there? No? Okay. So there are several ways to tackle these types of pathogenic protein aggregates that have been targeted. Reducing production, which is actually something that big pharma and biotech are doing a lot of. They're looking at A-beta modulators, and they're also looking at vaccines as well against A-beta. But they're particularly looking at reducing production of A-beta to try and slow down the production of A-beta plaques. So far, not successful, interestingly enough. So that's where some of the people who are against the A-beta argument kind of bring up the fact that we've been focusing on A-beta probably for about 10 years, and we haven't been able to develop anything that effectively slows down Alzheimer's. Even when you clear a lot of the plaques, you get things like brain inflammation, and you still have neuronal death. So it's an interesting one.
Wasn't there a drug that was tried to sever these cross-links, but then it actually caused brain hemorrhages, and a few people died, I believe?
Yeah, I think so. And I'm not sure if this is linked with a vaccine, because there was a vaccine that was being developed by Johnson & Johnson and Elan. I don't know if that rings any bells. And that actually was a vaccine against A-beta. So it was basically using the immune system to clean up the A-beta plaques.
That is, like an autoimmune response or something.
Exactly. What ended up happening was they got inflammation in their brains, and you got the hemorrhaging. And I believe some people may have died as a result.
If I may chime in on that.
Yeah, please do.
I remember the results of that study well. So there were basically 3 vaccines that announced their phase 2 and phase 3 results, and this was Elan's active vaccine, and then the monoclonal, and then Eli Lilly's monoclonal.
Okay.
They all showed the same thing. So the vaccine had the side effect in 6% of people that had some inflammation, and those were scratched from the analysis. But the other guys, this was in 2002, the other guys are now dead, so their brains got chopped, and we could see what actually happened in great detail. And what we saw there was that both the active vaccines and the monoclonals cleared out A-beta in a lot of patients, like undetectable, nothing left, completely resolved the plaques, and those were the patients that died with end-stage dementia.
Okay, so there we go, right? So the A-beta...
I mean, I don't know why anyone's talking about A-beta and Alzheimer's disease after these results, right? We have some mice that behave differently, but, what's the better model for a human, a mouse or a human?
Mm-hmm. Yeah, agreed, John. I mean, it was, to me, when I was working with Big Pharma in particular, and I'm wrapped up in a bunch of NDAs, so I can't mention specific names, but they were all still working on A-beta therapies, and this was as of a year and a half ago, right? So it's an idea that hasn't yet died, and I just didn't see as much work going on in tau, which is also, I mean, tau is interesting. I know that there is some work going on in tau, but it's an extreme minority. Go ahead.
Thing, by the way, in these Elan vaccine studies, they also looked at tau in those brains.
Okay.
And the tau is not gone, but strongly reduced. in the patients that died with end-stage dementia.
Okay. Another scratch. I kind of like tau. I like the story of tau that kind of gets in there and little subunits do a little something, but who knows? So as many of you are seeing, especially drug development in humans, there are a lot of unknown unknowns. Many of you already know that, just having experience in science in general, and especially preclinical models. But there are a lot of unknown unknowns. So we're still kind of rummaging around in the dark in many of these diseases. There are also some other hypotheses around Alzheimer's. So there are things like type 2 diabetes that may actually be somehow involved. So here we go. I just got a little something here. Here we go. I got a question. So Kelly's microphone isn't working. Are prions intracellular or extracellular proteins? They are. They definitely exist outside the extracellular space. I'm not sure if they operate in the intracellular space. I would assume so too. So I think they exist in both places. What types of research protocols are currently used to study them? Is there any interest in being able to study intracellular protein changes at the single level? I think most definitely. I think if we can do that, so I'll just repeat that. Is there any interest in being able to study intracellular protein changes at the single level? That would be great if we can get something that's accurate at the single cell level. And Eric's just responded as well. The big issue is the single target approach. So he believes that going for one target isn't enough. That may be the case. It's hard to tell. So, John, do you have any other thoughts on some of the other pathologies? So, particularly when we're looking at alpha-synuclein in Parkinson's, which is interesting, and TDP-43 in ALS.
Sorry, don't know anything about those.
Okay. And I know that the research is still, it's still very recent on alpha-synuclein. With the Michael J. Fox Foundation, we ran their studies, I think it was about 3 years ago, and that data has been published. And it showed the association between alpha-synuclein and progression of Parkinson's, but we're still figuring that out as a biomarker. With TDP-43, it's even more recent. Eric, I don't know if you have any thoughts on TDP-43. So for everyone just to know, Eric Valor is a friend of mine, also an ALS patient as well. Eric actually is communicating with us just with one eye. So he's actually on a full respirator, fully quadriplegic, and communicates just using one eye. He uses a type of eye gaze software to communicate. He's actually a pretty fast typer. He's kind of like a slow to medium typer, just with his eye, one eye. So I don't know if he has any thoughts. Eric's been doing a lot of research on TDP-43 as well as some other mechanisms as well. Adam, did you have any thoughts on TDP-43? I know you've done a little bit of research.
Yeah, I actually, unfortunately, I haven't really looked at TDP-43 a lot. I've been kind of run for a loop. I've been pulling some other papers with some researchers on topics not quite related to the curcumin studies, so none of the non-target studies. Stuff that looks interesting but still needs more evaluation.
Okay.
So
yeah, and that's useful as well. So the curcumin study, we actually are, just for everyone else to know, so one of the drugs that we're actually looking at developing, and we've been told now will actually be tested in SOD1 mouse models, which SOD1 is superoxide dismutase 1. It's actually one of the genetic models for ALS. 95% of ALS, we don't know what causes it, but there may be 5% of cases that are genetic, and so SOD1 is one of them. And we are actually, over at the Salk Institute, we are going to get J147 tested to see if it actually has an effect in protecting motor neurons, reducing protein misfolding, and also reducing inflammation as well. The mechanism of action of this molecule is not known, so it's something that big pharma and biotech don't like.
Ryan, just so you know, I actually did come across a paper. Some researchers at Brandeis have come up with a molecule that binds to the cysteine interfaces, the cysteine residues in the SOD1 homodimer that keeps it from dissociating, and the dissociations a major part of the aggregation. So that looks very interesting.
Okay, that does look very interesting. Does anyone have any additional thoughts? And actually, so that links in with, I know we kind of just started on that path, the therapeutic aspect. So there's stabilization as well. And we know that at least intracellularly, cells actually do stabilize their proteins. So there are lots of chaperones intracellularly. So that's another interesting angle for a therapeutic approach to neurodegenerative diseases, and particularly these protein aggregates and disrupted proteins, and then also disrupting formation as well. We do this regularly for certain things like antibiotics and bacterial cell walls. Right? We attack the polymerization of their cell walls, and we can unravel and kill those bacterial cells. So there are multiple approaches. I think right now it's still pretty much a very new area. Other than the old models that big pharma mostly is still holding to with A beta, there is some innovative work going on in the university labs. It's still very early. So does anyone have any comments on Alzheimer's, Parkinson's, ALS, or any of the other neurodegenerative diseases? Any questions? We have a lot of scientists here, so a great place to bounce ideas around on.
I have a question about well, it goes back to the amino acid sequence. I don't know why I'm so stuck on this, but are they protease resistant primarily because of their structure?
Yes.
Or are they protease resistant? Like if someone were to engineer, because there is a locus, a gene locus. I'm sure there are many other factors as well, but at least like with the PRP, there's a gene locus, right, that has been shown to be the producer of these prions or prions. I don't know. I'm mispronouncing it, I'm sure.
I was mispronouncing too. Don't worry. I have the American-British thing going on. So
if one were to maybe, again, this is assuming there are no other factors involved, which I know is not the case. But if one were to maybe engineer that site in the chromosome, so I don't know, I just came across it. If one were to mutate that site or put some protease degradation sites or something into these repeat regions that, could you increase the chance of these proteins to be degraded by endogenous protease is all I'm asking. Or is that just...
That's an interesting, so you're talking about using gene therapy to insert?
Maybe something along those lines.
Yeah, I mean...
but I don't know if it's because they form these it's either you put protease sites in there or you disrupt the structure so much that when they fold, they don't make these repeat units. But I don't know. I'd have to look into the element more. I haven't spent much time looking at it yet. But
I think from the paper here on page 46, they mention in the middle of the right side of the page that, like prions, A-beta seeds range in size from small, soluble, protease-sensitive aggregates to large in size. So I think it's more of a structural thing, that when they aggregate too much, then it kind of hides all of the protease binding spots or whatever. So I think it's more the structural.
Okay.
Yeah.
But again, then disrupting those small structural units somehow, maybe at the genetic level, if it is, because some of the diseases are, it's arise from mutations, right, in genetic elements and things along those lines. So I don't know if anyone's developing or looking into that means it's a therapy, but.
If you are talking therapy, you have, of course, the delivery problem, right?
Yeah.
You can't put it on the genome and change something. So I would go the other. Of course. I would start with engineering the protease to work better on those things.
Yeah,
right?
sure. But even if you could hit like a large there are many AAV vectors that are now being used in the brain. And of course, they're not hitting all the neurons or anything like that, or glial cells or whatever populations we're talking about. But if you could if you know someone's going to, this is a whole other thing. So if someone's predisposed to developing, Alzheimer's via prion disease or something like that, then maybe targeting a population, or I don't know, like maybe we don't have to hit everything. Maybe we can slow the progression. You know, like I'm not really sure, but you are correct that you're not going to hit. It's a delivery problem. You're not going to hit every cell, and that's like, you know, all of gene therapy's woes. But still,
I mean, you can probably publish a cool paper if you make a transgenic mouse that does that, right? Because in those mice, you can actually cure Alzheimer's that way.
Yeah.
Well, John, remember, if you know what causes Alzheimer's, which right now we're still at the drawing board, right? We're kind of looking at the drawing board and going, well, A beta, scratch, tau, probably scratch.
Yeah. Well, A beta certainly causes Alzheimer's in A beta overexpressing mice.
Well, yes.
Yeah. And a question on that. In the study with the vaccines that eliminated the A beta, I assume that was late stage patients. And is it possible that kind of the damage was already done?
I believe it was definitely seniors. John, do you remember the age ranges for those?
Well, it wasn't bound by age, it was bound by Alzheimer's stage. And yes, two of the studies were late stage. One of the monoclonals was mid stage. And yeah, that's what the press release said exactly, is that the damage was already done, and now we need all this government funding to run a study in early stage patients. So sure, you can always weasel your way out of that, definitely.
One thing,
a different class of drugs that were intended to slow production of beta amyloid by targeting secretase pathways and things like that, right? And about those drugs, I don't know of any autopsy results from those drugs, but I do know that all of them were, of course, completely inefficacious. So maybe the way to go about answering your question would be to look at drugs that slow down beta amyloid production as opposed to clearing existing plaques.
It's a good point. I would say it's a very good point that in drug development, generally the first people to receive a new drug are going to be patients that are far along in the progression of a disease and have run out of other options, particularly where there's a standard of care. So it can be extremely challenging if there is an existing standard of care to develop a competing medicine.
Yeah.
Well, there is.
I believe they discuss this trial or the trials of these drugs in depth in one of Aubrey's books, Ending Aging. And one thing I think I might remember from that is one of the hypotheses for why beta amyloids might still be involved in Alzheimer's, and why this drug that sort of cleared up all the plaques may not have been the solution, is that I think they theorized that perhaps one of the intermediary phases of aggregating may have been more toxic than the fully formed, beta amyloid sheets or whatever they call it in its final form. So I think that the drug might have broken up the large aggregates into more of the intermediate phases, which in turn was more toxic. So I think that was one of the theories of why maybe it didn't work. If that makes sense.
Not so sure. I mean, the drug was a vaccine, so it should result in these things being taken up into macrophage lysosomes, and there shouldn't be much left after that. But yeah, I don't know.
Yeah, but it's a good point, and that goes to the actual disruption of the cytoskeleton of the neurons. I think they were like tubulin or something, that those precursors, those A beta precursors, and possibly also tau precursors, may actually disrupt the function of the neurons and kill the neurons. So it may be toxic before it becomes a plaque. I think that's was that kind of what you were referring to? Yeah.
Yes.
Yeah.
So that by preventing the formation of the the full-on aggregates, it was actually holding things in the more toxic phase.
Exactly.
No, if you're talking toxic oligomers and monomers and stuff like that, those were gone in those patients.
Then I'm not sure if I'm talking about the same drug trial links. I'm not sure if this was a vaccine, more of a breaking of the aggregates that were already there. So maybe it was a different drug.
Yeah, because the aggregates could be the brain's way of safely keeping away toxic substances from itself. So they form the big plaques so that it's not soluble in the brain.
Right, yeah.
Yeah.
That was the theory, I think, that I'm remembering from that book. I'm not sure if I'm remembering it correctly, but that sounds very close to what I'm recalling was their explanation for why it might not have worked.
Yeah, there are many theories, and there are even when you start to look at really closely, even at A beta, there's an incredible amount of complexity. So A beta isn't just A beta. A beta is composed of several different subunits, A beta 42 and various other versions of A beta, right? The actual subunits to A beta, and so it's a very nuanced discussion around the structure of the A beta itself and the biochemistry. So there were companies that still believe, small biotechs that still believe you can actually still work on A beta and have an effect, that I've worked with in the past. So yeah, so I don't know, any additional questions or comments? Or now here's an interesting point. So they've said that this is the decade of the brain, but we're still moving pretty slow on the brain. There haven't been a lot of innovations yet in treatments for CNS disorders. The last major innovations really were the antipsychotics and the atypicals. That was really something that made a difference between night and day. I mean, you had patients who were locked up in insane asylums, and after treatment with the antipsychotics and atypicals, were able to actually leave and function at least moderately normally. So that was a revolution. Undoubtedly, with some of the basic research that's going on and some of the GWAS data that we're figuring out, hopefully we'll be able to come up with better therapies. So we're still figuring that out. I know that Eric and I we've got our fingers crossed that J147 works out. Unfortunately, we've seen cases where it hasn't. KNOP is a good example in ALS. Dexapramipexole looked really, really good in phase 2. In phase 3, looked like it didn't work. So it's tough. Drug development is tough, and it's not just because of the science. It's also selection of the patients, and it's actually what Matt was indicating, which is that the sickest patients usually get treated first, and this can be a really big problem because oftentimes a lot of the damage is already done. So it's like waiting for in oncology with the studies that I've run in oncology. You wait for someone to have failed 2 or more therapies. So by the time they're very, very sick, they've got metastatic cancer, it's spread to pretty much everywhere. Now you treat them with this new drug. And then how do you know if this new drug may have worked as a first line?
This is why Michael J. Fox has actually changed a lot of the focus of their funding right now, away from clinical trials to biomarker research, because they put so many millions of dollars into it. They have in the last 2 years, I think, they've been doing a $40 to now almost $60 million initiative to find biomarkers to better model the disease so they actually understand what they're trying to target, rather than trying to hit targets for end-stage patients. Let's catch the disease earlier, let's understand it earlier, et cetera. And I think that and the way they're using stem cells to model disease are going to be very interesting.
Yeah, and that's a really good point, because, I mean, Michael J. Fox Foundation, I think, is one of the pioneers in actually, so their motto is they're not just doing research, they're looking for a cure. And I was involved in helping them design those early phase studies that were looking at biomarkers. So we looked at that, and now this is all public information now. They've published this data. But we were looking at the cerebral spinal fluid of patients, and then looking at alpha-synuclein and various other biomarkers. They haven't published the other stuff, the other biomarkers yet, I don't think, but they were looking at various other biomarkers to establish the disease, the course of the disease, and then move on from there. Now that we know the biomarkers are involved, how do we shift those biomarkers down? So it's in humans, so we actually have real models rather than just saying, well, in animals it looks like this, because there are no really good models for that. So
biomarkers are really important, and they're a good target for DIY scientists, particularly if you're suffering from a particular condition and you must have the biomarker in you somewhere for a condition. So yeah, it's a good target to go for.
Yeah. So, and we're still figuring that out. I mean, Alzheimer's, I guess if we're being honest, we don't really know what the biomarker is right now. Yeah, really. So how can we treat a disease that we don't know what we're trying to affect, other than hopefully protecting the neurons? So
there's one thing we know about Alzheimer's, which is ApoE, of course, right? The gene that can pretty much guarantee you Alzheimer's disease by age 80, or or it can guarantee that you never get it, depending on what type you have. Right. So, I mean, if I have that strong a pointer from nature, that's kind of where I'd be looking in terms of both markers and cures.
Okay. And that maybe we should look at that. ApoE4, maybe that's something we should discuss in further detail as well. So I've written that down. That could be a new topic. So okay, so we're kind of getting close to the hour mark. I don't know if actually we do have one more question from Yurli. The in-teams. Do the in-teams have much of a role in such protein aggregation? Make it more comparable with the template seed. I don't know if anyone has any thoughts on that. In-teams.
DEX is another classic single target failure. I still believe it could be effective in a multiple target treatment.
Could you repeat that, Eric?
DEX was another classic single target failure. I still believe it could be effective in a multiple target treatment.
Okay, so dexapramipexole was the Knopf drug for ALS, and that's the one that failed in the phase 3 study. Didn't meet its particular endpoints, but Eric does feel that, and actually to some extent I do too, that it still has potential as a therapeutic. Just having looked at the fact that there were some responders. So this is the challenge with drug development, right? It's the devil's in the detail.
Yeah, that's where the biomarkers and genotyping really begins to help you, because if you can correlate a particular genome with a sub-patient population, then you have a shot at saying, well, I'm going to make a test. Just enroll people who pass the test.
Yeah, and that's where we've started to go with certain personalized medicine like oncology, where we know that there are definitely like BRCA1 mutations and BRCA2 mutations. We know that there are some cancers that are more responsive because they're BRCA1 or BRCA2. So we can go after them. But I don't think we're there yet, unfortunately, with many CNS disorders.
Yeah.
Yeah, unfortunately. But hopefully the GWAS data shows us lots of really, really cool stuff, and then we can start going after that.
Well, we do have that a little bit in Alzheimer's. From our last trial, we had a subgroup where the vaccines, the monoclonal was efficacious, which was ApoE homozygotes, right? And Elon said, "Have you heard of Mr. Bonferroni? We're out of here." And the government is resurrecting that though, and they just dumped $5 million of NIA money into repeating that for that subgroup.
So John, the message that I'm getting here is we need to look at ApoE4, right?
I don't know. Elon thinks that this is a statistical fluke. The government thinks it's worth throwing my money at.
Oh. I don't know if many of us are convinced about the government knowing what to do with money.
I don't know much about ApoE4. I know it's a strong correlation. It was done. It's a SNP correlation. Is that actually a mutation that, is that actually a gene that has a known effect, or is that just a SNP that they don't know what's going on? It's just correlated statistically.
Well, we don't know how it mechanistically relates to Alzheimer's disease. We do know what ApoE does. ApoE is an HDL carrier protein, right? It makes the good cholesterol particle, the HDL. And so people looked a lot about what it does in heart disease because of the cholesterol in the arteries. People don't really know what that has to do with Alzheimer's disease mechanistically. And it's an interesting question, and somebody should study it, but nobody does because these protein aggregates are so much more hip still.
And is that SNP, is that change actually in affecting ApoE, the actual gene itself, or is it just like it linked to it in the area? Is it actually affecting APO itself?
Yeah, they are two SNPs with effects on the amino acid sequence. Yeah.
Okay.
Yeah. And just to give everyone a little bit of information on that, the Caucasian carriers of 2 E4 alleles have between a 10 to 30 times increased risk of developing Alzheimer's disease by the age of 75. So it's significant.
Right, and they couldn't have any more fold increase because everyone has it at that point.
That's a good point. Yeah, I think what is it, John? It's like a 50% risk of getting Alzheimer's.
No, if you have E4 and you're 80, you have Alzheimer's. End of story.
Okay. No. But I mean in the general population, we just take the American public.
It depends. It's highly age-related, of course.
Yeah.
So pick an age group.
Okay. All right. The old age group, the let's say 70 plus.
Like the best way to avoid getting Alzheimer's disease is to die from something else first, and that distorts the numbers. So you have to say, I'm looking at 85-year-olds.
Okay. So there we go. So maybe that's something worth looking into, and that's really interesting because HDL. Maybe there's something more to the story of HDL than just cholesterol. You know, and cardiovascular disease, or maybe actually that's a good point. Cardiovascular disease that could be linked with Alzheimer's, right? I mean, if we're talking about clogging of the blood vessels that supply blood and oxygen to the brain, that could have an effect.
Actually, I have a question now that you're mentioning cardiovascular disease. Mm-hmm. I'm just wondering, so far, if researchers during developing models to understand prions, has that been understood somewhat closely to the way, say, cancers are, that we try to understand them, or is it considered more closely to, say, a virus or some kind of phage, because I'm wondering, how does that in turn influence the kinds of therapies and the therapeutic approaches we're developing on prions in the first place?
So prions, when they were discovered, were an entirely new disease vector, right? That was something I actually remember. I think it was discovered around the time that I was finishing my first degree, and it was just very confusing to people. So I was actually in the UK. This whole mad cow disease thing blew up. Actually, it was before my first degree, but it was an entirely new disease vector. It was not a virus. It was not a bacteria. There wasn't even a eukaryotic cell type. So it's new. It's its own class of a protein, an infective protein disease.
Okay, that's good to know. Because I was talking about vaccines being developed, and I'm thinking maybe it's just somewhat shaped the way that we think we can cure it. But if it's just its own real thing and it's baffled everybody so far, then there's still room to, make our own kinds of new relationships and new links between that and some kind of novel form of therapy that we wouldn't have thought of necessarily.
Mm-hmm. And there's incredible potential for that, right? If we start looking at, and actually what John was suggesting, like proteases, why don't we start looking at proteases that break down normal A beta and start mutating them and see what happens, right? Maybe we'll get something that will break down if the A beta story is true, and it looks like it may not be true. But using that approach to the other protein aggregates.
So one of the hypotheses for prions that I've heard is that it's kind of a self-replicating molecule that the prion actually catalyzes the formation of other prions, and that's how it reproduces and expands in the body. So, from my perspective, I think that maybe some work with basic self-replicating systems, the sort of chemistry that was very popular when I was in graduate school was, being able to develop these self-replicating molecules that, kind of acted like a machine or DNA. And it might be interesting to see, are there ways that can be interrupted, that process?
Mm-hmm. And that's something I can look into it for us, so that maybe next time we can have another review of a paper that's looking at how to disrupt things from a chemical perspective, right? Like looking back at some of the older techniques. Or Matt, if you're interested, you want to look into it.
My brain is churning now, so
Okay.
It'll ping me.
It'll ping you. Okay. So Nina, remember that there may be a little journal club with Matt leading it, looking at some of the chemistry techniques to attack these.
I keep a mental list of people I'm going to solicit. So never. Once you mention an interest, you're on the list.
All right. So I think we're basically we're pretty much at time. It's about an hour. Does anyone have any other questions? Yeah, please do. Yeah.
Along those lines.
Okay.
I'm not a subject matter expert, so I'm not sure. But one thing that caught my eye is that it's a crystallization like chain reaction, and I'm wondering if anyone's looking at that. How what kind of pattern it follows? Is it dendritic? And if not, what the obstacles are, because I looked at figure 1, where it showed the commonalities among age-related diseases, and it said that it's actually these, the model was inferred from patients. It was post-mortem, so it's an inference. It's not even an actual brain scan.
Yes.
And so I'm wondering, is it possible to do these brain scans right now and be able to see how the plaques develop and aggregate?
That's a good question. So I do know, and John, you may know a little bit more about this than I do, but I do know with MRIs, I think we can look at the plaques, particularly when we're looking at Alzheimer's. With the others, I think it's much harder. So if we're looking at, especially the intracellular, when we're looking at Parkinson's and the Lewy bodies, and we're looking at TDP-43 and ALS, I think that's much harder to do. I don't know
I don't think that MRI is capable of imaging down to the cell level.
No, not really.
No. You need like a 14,000 Tesla MRI.
Yeah, right now for Parkinson's, they only diagnose it when post-mortem, you can only get an accurate diagnosis that you had Parkinson's. So if you get your brain autopsied afterwards, other than that, it's just conjecture that you responded to medication. So that's probably what you had.
MRI is not really that sensitive a technique. It does do gross features of the brain, but it's nowhere near the cellular level.
I think they were working on new PET scans for Parkinson's that they were testing out to see whether or not, as part of the biomarker initiative Michael J. Fox was doing, using a new, I don't remember what it was. It was something new that was just getting FDA approval that would be able to maybe show it. But those Lewy bodies are so tiny in such a tiny portion of the brain, you don't see anything. Yeah, I mean, at best you'd be able to have a heavy metal targeted antibody or something like that to bind to the structure, and then you would be able to see, oh, this area or the other has more or less of it.
Great. All right. So I just wanted to thank everyone. Actually, this has been like a super conversation, really, really interesting, and I think that the real values really come from the conversation and discussion from lots of great scientists and lots of great minds. So I wanted to thank you all. And then I guess we'll be doing more interesting idea labs. I'll probably be focusing mostly on therapeutics, as that's one of my main areas of interest. But thank you everyone for joining, and thank you, Nina, for setting it up. Nina, quick question in terms of, I know we're recording. Where is this being shared?
We're still working out a good place. We've recorded all of them so far, and I think we're still trying to find the best place to put them.
Can you get them up on YouTube?
Yeah, something happened. I think Derek looked into it, and I don't remember the end result of that. But is everyone okay with sharing these on like a private YouTube? Something happened. Like we wanted to share them on a private YouTube channel, and I don't know what came of that. So I think we have to look into it a little more.
I just think it would be really valuable. I know there was a lot of interest.
Yeah.
Yeah.
I had another science question. I'm sorry. You may have mentioned this, and I completely apologize if I spaced while you guys are talking about this. But what about a lot of small molecules are being developed to disrupt virus particles, right? That have like repeating monomer structures, whatever. And, you know, maybe something similar could be developed to disrupt the subunits that form these long fibrils or amyloids or plaques or whatever. And I didn't know if there's a lot. I think there might be. I think actually there might. Now that I'm mentioning it, I want to say that there have been some small molecules developed to try to disrupt prions, but I don't know if that's being continued or what the end result of that was, or if anything's gone into clinical trial. So I don't know if anyone can comment on that.
Let me have a quick look, because I do remember people working on ways to disrupt prions. First treatment. Okay. So they are currently working on it. Researchers in Sweden are working on something.
Okay.
Yeah. Yeah, it's actually an anti-malarial compound that appears to reduce the growth of abnormal prion proteins, and there are a few others that look interesting too, including a derivative of beechwood, actually, beechwood shavings. Interesting. It's just stopping the growth. So tetracycline, glutarathine, and antipsychotic, actually. No survival benefit was seen, so that doesn't work. No. So there's definitely treatments being researched. I don't think anything's been approved yet, though.
Okay.
What is that? To start drinking beechwood tea?
Yeah, looks like that might be a good thing, just in case. And of course, not eating anything with brains in it. That's usually. For full disclosure, I am vegan now, but I ate lots of hamburgers when I was younger, so I need to be careful.
I was going to say, I'm very paranoid about pesticides in a way I never was before my last job.
Yeah.
For the record, I still eat haggis, so.
Okay.
If you see me keel over.
If we see you shaking and your brain swells, we'll know what to do. All right, everyone. Thank you very much. This has been a pleasure, and really enjoyed talking with all of you, and look forward to joining the next Idea Lab.
So what are we doing? Do you want to discuss? We actually discussed the topic last time, and then we didn't stick with it. So,
are we leaning towards ApoE4 Alzheimer's or not? Or, I don't know. Is that a topic or?
That would be.
I mean, I would be interested in learning more about it. If you guys are willing to. If someone's willing to present.
Anyone willing to put their hand up and do that one?
I don't know anything about it, so I would.
Yeah, I know nothing.
That means it's you,
I'll pick the paper, sure.
There we go.
All right, John knows the most about it.
That's our guy. All right. All right. I'll join you over in the lab, John, when you're presenting. How does that sound?
Yeah, send me something in writing so I remember it.
I will. I'll send you an email.
So I'm looking at the paper that just came out of UCSF on telomere length, and I'd like to do a paper on p-values and error bars and why they can make a really good result look really bad.
Okay.
So I think, I kind of want to debunk that paper. And I'm sure there'll be plenty of people that want to disagree with me on that. So.
What paper is this? Telomere paper?
Let's see.
Is this the one with the exercising makes the telomeres regrow and all that?
This is the one where they intervened with prostate cancer patients and made them make healthy lifestyle changes. Yeah. One group healthy lifestyle, one group continue on, and the healthy lifestyle group got an increase of 0.07 TS units, which, if my math is correct, that's about 2 years of additional life.
That's bad in cancer, isn't it?
But the error bars on that, based on my calculations, are plus or minus 3 years of life. So
how did it get published?
Well, they actually only published the correlation between the study and the TS levels. And TS levels, you have to go through 2 analytical methods to get to actual, to get it back to the original study. What is it? Carruthers? Can't remember the C name. That actually did the correlation between biological age and length of telomeres. So, and that correlation, actually, they reported it as R instead of R squared. So they have an R of -4, which is an R squared of like 0.16. And that would be so far an F in my analytical classes that, I can't believe that, they actually published a result that had an R squared of 0.16.
I think John has a comment on things being published. John, you want to chime in with that?
What? Why did that get published? Was that the question?
Yeah.
Well, this kind of stuff gets published because, the kinds of people who go into biology in academia are the people who kind of like nature and aren't so good with math, right? That's why you pick a bio degree. And those are the reviewers.
So maybe we need a statistics lesson next journal club.
Well, I can't do one for the next class, but I'm working up the slides for a future class. So Nina, you can put me down for that if that's interesting to people.
Yeah, definitely. Yeah, add some critical thinking.
Yeah, I think it's maybe a skill that's not commonly taught. In a lot of discipline areas. I know you can't get through a chemistry degree without, some basic statistics. I just don't know what exposure people have to it. And, from a citizen science perspective, being able to critically think about a paper like this and follow the train of papers back to the original analytical technique is helpful. On the other hand, it'll probably bore all the senior scientists.
I think we all need a refresher.
Yeah, absolutely. Oh yeah.
All right. So I think and it sounds like you're getting a nice long list, Nina.
I do. I have like the next 3 planned out already.
So are we wrapped up?
What about so the thing that we were doing really is like alternating between, well, now it sounds like a battle, but we were alternating East Coast, West Coast with
all right.
presentation.
Put your gang signs up, right?
So I don't want to. So yeah, obviously, GenSpace folks, chime in if you want to do we want to stick with that. Like you guys want to lead the next one or something. I don't know. Or do we just want to make it like all participants and we just rotate through participants in the journal club? Since more people.
I don't know if we have a format. I mean, either one is good for us. If,
okay.
Or we could just rotate around.
If that works for you, that sounds good to me because more and more people seem to be joining. So.
Yeah, I mean that's probably better because sometimes if there's only like one person that shows up or 3 people on our side, then it'll one side will kind of dominate and we'll just keep going back and forth. So it seems like we have a huge group here and growing. So I think we have a lot of room here for the squares to kind of rotate around. And it sounds like somebody's already picked an Alzheimer's paper. So yeah.
Sounds good.
All right, and Nina, so are we going to hopefully see some of these up soon on YouTube? Derek?
Yeah,
Cool.
we'll figure something out. I think obviously we can't do Dropbox because it will take up too much space.
Yeah.
And there was some issue with the YouTube, wasn't there? I just don't remember what it was.
Yeah, the issue I had with YouTube is that it wouldn't allow me to do recordings over 15 minutes, I think. And it may have been something with my account, but I wasn't able to get it to upload anyway.
Usually if your account is not like the first thing you've ever done on YouTube, you will get more than that 15 minutes. I haven't really known anyone to have that limit anytime recently. So if it is a limit, just post like 2 normal videos or something and don't do anything illegal and you'll be extended.
Ah, okay. So I was trying to create a new account to host these.
It might have been like a probationary period kind of thing.
Gotcha.
Okay.
Yeah.
All right, well, we will attempt that again then.
Yeah. I mean, you could do 2 things when you do post a video. You can either make it a private link that anybody can see as long as they know what the link is, or like you actually like whitelist particular people that can see it. So you can do it either way, whichever you want.
Well, let's get some feedback from that. I don't see any reason not to make this anything but a public link. Does anybody have any concerns around the privacy of the data on this call?
I think that would be great.
That's fine with me. Yeah,
cool.
Yeah, I'd love to look at the ones that I missed, you know, the old Idea Labs, so that would be great.
Yeah.
Would it make sense to also sort of compress it down, make it maybe turn it into a slide presentation? Rather than having full video the entire time, or a podcast.
Possible.
Yeah, we could do something like that, just audio.
Yeah, just strip out the video, leave the audio, and podcast it and call it the DIY Bio Idea Lab podcast.
That's not a bad idea, actually.
Yeah, that's going to be a lot more post-processing. It'd take quite a bit of time to prepare that, I would think.
It is. I also kind of like the effect. I also kind of like the effect in the recording of having the face of the person speaking come up. But I think even in the recorded ones, it will tend to add something.
Let's make ourselves cartoon characters.
They don't have to be mutually exclusive. You know, you could post up the videos on the YouTube channel and then strip out the audio and make it a podcast. So it doesn't have to be one or the other.
Agreed.
And the Zoom platform is superb. It's the most stable video platform I've ever seen.
It's working great, isn't it?
Yeah.
It's awesome.
Google's ass.
I felt like I was in the future, walking along on the street on the bus on my Android phone, still able to record the video. That's fantastic.
That's awesome.
Or like Matt, who was driving last time.
You don't drive in CAS. That's dangerous.
All right, guys, I got to take off. Actually, I got to be at Keith's house.
Bye, everyone.
Bye.